Okay, let us get started. It's 9 o'clock. I like to stay right on schedule, so we're going to get started here. Dr. Karl Lintner is from a native of Austria. He studied chemistry at the Technical University of Vienna, and he obtained a degree in chemical engineering, where he focused on the domains of pharma and food biochemistry. After a long career in industry, in 2010, he set up his present consultancy company, Peptide, which allows him to participate both in ingredient development and advising finished product formulators in many aspects of cosmetic research. He also has teaching positions at the ISI-PCA Institute and at the University of Versailles. He is a member of the Society of Cosmetic Chemists in the United States and a member of the SFC and also a member of the DGK. Those are in France and in Germany. Germany, I think, the DGK.
Yeah.
Okay. And he's a part of other organizations. In 2012, he was awarded the prestigious Maison de Navarre Medal of the SCC, which is the highest award in the SCC. And in the spring of 2013, he received the In Cosmetics Lifetime Achievement Award. So we'll get started with the slides here. Welcome to the presentation, Karl. And let me start the slides up here for you. And we'll get ready to go. Take it away, Karl.
Okay, thank you very much, Perry, for this wonderful presentation. I'm all moved. Thank you very much. Ladies and gentlemen around the world, it is a pleasure to speak to you today. Thank you for attending this seminar. I'm allowed to speak about my favorite subjects, which are peptides, and especially, of course, peptides in cosmetics. Now, why? Why is peptides such a topic that we get almost 200 people together to listen to this? Because peptides made a breakthrough in the market over the last years and have become a major staple in cosmetic products. Why? Well, because, as you shall see, they are something special. Special in their activity and special in how they're made and what they do. It turns out that one of the peptides that I was happy to contribute to and to participate in the development received in 2015, I think it was the 25th edition of the In Cosmetics, and at this time a peptide of our development called Matrixia won the Innovation Award, the 25-year Innovation Award, which recognized the product that had the greatest impact on the personal care industry in the last 25 years. So that was a wonderful recognition of the importance of peptides in cosmetics. But I'll stop here with lauding myself. Here are documents that show why peptides are so interesting, because they're truly active and active without any side effects. Here you see the wrinkle reduction, non-photoshopped images, the true images, where you see the improvement in skin features, wrinkles around the eyes, and even wrinkles on the front. In vehicle-controlled randomized studies, you see the benefits of peptides in a cosmetic product. Or with pictures like this is 2 months. And the interesting thing, as we shall see, with very small amounts, 3 ppm, that is 3 milligram of peptide, of the pure substance, in a kilogram of cream. And you can achieve wonderful results. Not only on wrinkles, but also on skin elasticity, for instance. This is with a different peptide, where you show the extendability of the skin of an elderly lady before and after using a peptide containing cosmetic cream. And the skin becomes much firmer and elastic and less toxic. And there are many more examples of peptides in this way. We don't have the time to go through 50 different peptides, but they're all very versatile and very active. Now, the agenda of today's seminar is given here. A little bit of history, a little bit of chemistry, mostly biological function, the use of peptides, and then some technical aspects, also safety and regulatory, and we come to a quick conclusion. Here's another document. The Dermatology Times recognized these peptides. That was a study by Procter & Gamble. Prestigious study and prestigious company. The first peptides were discovered in the 20s, 20s, 30s, 50s of the previous century. Glutathione, which most of you may have heard of, a tripeptide that is circulating in our body, antioxidant peptide. Insulin was discovered in 1947, much bigger peptide with 54 amino acids. And then it really started to become interesting when scientists discovered small peptides other than glutathione with very specific activity in the human body and in some animal bodies, mammals, pigs, and others. For instance, oxytocin, which is a peptide with 8 amino acids, and that is called the happiness or the hugging peptide because it induces labor for giving birth. It induces lactation and has other psychological benefits. Vasopressin is a peptide with 9 amino acids that induces, urinating, eliminating water. And at this time, people started to learn the composition of these peptides, were then able to synthesize them and to start a whole new chemistry of peptides. And here's a list of other peptides that are all in our body, circulating and playing very important role that is similar to other molecules in our body, which have what we call hormonal function. And so, indeed, when we think of hormones, mostly those are steroid hormones, the so-called testosterone, estradiol, sexual hormones, but peptides also are hormones. They're just messengers to deliver a message to certain targets in the body, cells or organs, to affect some activity. We won't go through the pharmacology of all these peptides, but know that there is a huge number of them, all of them important in the functioning of our organism. So what is a peptide? Well, I'm sure most of you know this from school, from higher education in physics, in chemistry or biology, biochemistry, but still, a peptide is a chain, a linear chain, no branching, a linear chain of amino acids which are coupled to each other. It's not just a mixture, they're added and coupled to each other in a specific order. The specific order, the sequence, as we shall see, is very important. And peptides can be a very, very long chain, but the simplest peptide is a dipeptide with 2 amino acids coupled to each other. And then we call it tripeptide with 3, tetra with 4, and so on and so forth. You need to know a little bit of Greek to understand this, penta for 5 and hexa for 6, and so on and so forth. In cosmetic application, it's rare that we use bigger peptides because there come questions of stability, questions of penetration, questions of cost and of toxicity. So most peptides used in cosmetics, with some exceptions, but most of them are what we call oligopeptides, oligo meaning a few, and that means less than about 20 amino acids in a chain. We talk about polypeptides when there are up to 100 amino acids, so that's already relatively big peptide. And beyond that, this is not the legal definition, but there's a general accepted difference or accepted border. Then they become proteins when they have more than 100 amino acids. Usually we speak about proteins, but again, there are some exceptions. You can have proteins that go to up 100,000 peptides, 100,000 amino acids, and a very large molecular weight. This is not the subject of our talk today. We'll stay with the oligopeptides. Here you have, which I'm sure you know, the structure, the chemical structure of the 20 amino acids, which we call natural. Of course, other natural amino acids exist also, but those are the ones that the genetic code uses to make our proteins. Only those 20 amino acids are found in our enzymes, in our collagen, in our elastin, in our proteins. And you can use these to make your peptides. Now, there are two categories of peptides, at least with respect to cosmetic usage. One is something that has been used for ages and was never something particularly original. That is the peptides that are generated by the hydrolysis of big proteins. If you take collagen, if you take wheat protein, if you take casein from milk, these are large proteins with long chains. And if you hydrolyze them with an enzyme or with an acid, then they break down into smaller fragments. But in fragments which you don't have the structure, you don't have the size, you have just an overall overall picture, let's say a fingerprint of what it is, but usually not very precise, undefined and unidentified mixtures. I represent this in an analogy with this pile of rocks. And then there is the peptides, those that have conquered the cosmetic industry over the last 30 years. Those peptides that are specifically designed, designer peptides, where you know exactly which amino acid is coupled to which, and you do this by synthetic means. And you have something like the pearl chain, where each pearl is an amino acid, and each chain, which has the pearls in different orders, will be a different peptide. So that is to be understood, that peptides from hydrolysis, well, it's a complex mixture. And the problem is, of course, every hydrolysis, every batch you make may be slightly different from one batch to another, because you can't control the parameters of hydrolysis down to the last decimal point. So there is no specific bioactivity. And in addition to that, sometimes you have odor and color problems, stability, and no real potency. To say that the product contains a collagen peptide, well, that's fine, but this is just something like this pile of rock. It is not a well-identified structure. What I call the real peptides, those that are of interest to marketing, to the consumers, and of course to the scientists, are those where you start with an amino acid and you make the synthesis in the right order. Here, for instance, of course, you can choose a peptide, a fragment sequence, a fragment structure from a natural protein such as collagen. You just break down the collagen, you analyze it, and you find a small fragment that may have some activity, usually or very often does. And for instance, here's a small peptide that you can find in collagen as a fragment of collagen, but you don't want to isolate it because that is not economical. You want to synthesize it. And here you have 3 amino acids, glycine, histidine, lysine, in this particular sequence. And that was actually the first cosmetic designer peptide introduced in the industry in the 1990s. Yeah. So how do you make a peptide in a reactor? This is an empirical science, but it is chemistry, peptide chemistry, very special techniques to attach one amino acid to the next. Between the 2 amino acids, what binds them together is what we call the peptide bond, is a bond between the carboxyl group of one amino acid with the NH, the amine group of the next amino acid. As you know, of course, an amino acid is characterized by the fact of having always a NH2 amino group on one side and the carboxyl acid group on the other. And by making this peptide bond, you eliminate the molecule of water and you get a very stable, and this is important, a stable bond that is not so easily broken compared to, for instance, esters. Now, how do peptides work? These type of peptides, these hormonal peptides, messenger molecules. They are produced in our body by various ways, from different glands, from different organs, possibly cut down to size by certain enzymes, and then they circulate in the body very far or very close to the place where they were released. And these peptides find a receptor, a cellular receptor somewhere that is specific to this cell and to the peptide. And when they fit together, then there is going to be action. There is mutual recognition between the receptor, which is usually a large protein embedded in the cell membranes. And when this recognition is solid, because they are precisely structured to each other, the receptor and the peptide, then something happens within the cell. And this is the simplified model of key and lock. When you have a lock, there is only one specific key that can open it, or one specific key can open only one lock. And so the peptide is the key, and the receptor is the lock. The ligand, as it's called pharmacologically, binds to the receptor, and then the receptor changes structure within the cell, and something happens within the cells as a consequence. So, for instance, here, when we talk about this peptide I mentioned at the beginning, that has an anti-age and especially anti-wrinkle efficacy. I'm not doing any advertising here, but just as an example, nature is very economical. Nature uses lots of material to affect what the body needs. Our bodies, as you know, they age. Sometimes they are also wounded by an accident or war or whatever. And as long as we don't die from the wound, we can try, our body tries to heal it. And wound research has been ongoing for decades and has found that in many cases there is a specific process that occurs that helps to clean, to repair the wound, to repair the tissue. Now, what happens? Imagine you cut yourself, you cut your skin somewhere in the finger or in the face when you shave, and you bleed. There is immediate reaction, there is stopping of the bleeding, and that's a complex process. Then there is a little bit of inflammation, which is in this case necessary and good, as long as it's just a little bit, in order to clean the wound, to clean out all the debris, the fractured and broken pieces of the tissue, of the skin or whatever organ. And then there is rebuilding of the tissue to close the wound and to repair it. So in this period of inflammation, enzymes, they break down the damaged, let's say, collagen in the skin, because collagen 1 is the major ingredient, the major protein of our dermis. Same thing with hyaluronic acid, which is not a protein, but it also breaks down during a wound. And the enzymes, they take this large damaged collagen and cut it into smaller pieces, which can be eliminated by the bloodstream because they are not wanted there. We want clean wound to build the tissue again. But a few of these small fragments that are generated by these enzymes in the wound healing process, they turn out to be active, to have a feedback activity. As I said, nature is economical. She uses even the debris, she uses the damaged fragments, and these fragments are recognized by the cells as signal of the wound. The cell sits in the dermis and does not necessarily know that there was a wound close by, but these small fragments of the proteins of the peptide, these fragmented peptides, they reach the cells and trigger the synthesis of new matrix molecules, macromolecules. Fibroblasts in our dermis, they are biochemical machinery to make HA, hyaluronic acid, to make collagen, to make fibronectin, elastin, and whatever the tissue needs. So this very simple process. Protein breaks down because of aging or wounding or some other destructive disease. These large molecules are cut into smaller pieces, each of a different type, but a few of these small pieces, small peptides, they turn out to be messengers for the fibroblasts in the vicinity, and the fibroblasts then say, hey, we have to wake up, we have to synthesize new collagen, new hyaluronic acid, new elastin to repair the tissue. And so this is what happens in our bodies, but this is what we exploit with peptides today in the cosmetics, because wrinkles are, as I say, the wounds of time. We get them because the skin is damaged, not necessarily with a knife, but with UV light and pollution and free radicals and so on. So when the skin is old, the dermis becomes less dense, there is less collagen, there is less elastin, and the wrinkles appear. Now, having identified some of these peptides that are active on the fibroblasts to do something, these peptides have been called matrikines because kinane is Greek, means to modify, to change, to move, to do something, and matrix, well, matrix is the extracellular matrix, we call it the ECM, is the tissue of our dermis, of our skin. So these matrikines are small fragments of these proteins. They are active, and if we synthesize them and put them on the skin, then if they can reach the fibroblasts shown on the left, then they can stimulate the fibroblasts to make new collagen, to make new elastin, fibronectin, hyaluronic acid, and everything else. And what happens? The skin becomes more dense and is repaired, and the wrinkles become less visible because the surface will be smoother, and the wrinkles don't disappear totally, but you can get, as we have seen one picture, but there are many of those, you can get some real fantastic results on wrinkle repair. And of course, not only wrinkle repair, peptides can do a lot of other things. So this is the principal idea of how cosmetic peptides work in the case of anti-aging, but of course in other activities such as slimming, such as elasticity firming, or skin whitening, or skin tanning. Other peptides have different activities, and not exactly the same mechanism as these matrikines, matrix repair, but simply interfering with certain biological processes, naturally interfering, because we use mostly peptides that we have in our body anyway, which is to add a few more to boost the activity, those activities that we're looking for. I have mentioned the word sequence before. Sequence meaning the order in which amino acids are attached to each other. If you take a peptide with 3 amino acids called tripeptide, there's 3, such as the glycine, histidine, lysine, okay? These amino acids have names, glycine, histidine, lysine, but if you have a longer peptide, it becomes difficult to recite the whole name. So you have abbreviation like gly, his, liz, for glycine, histidine, lysine, and even further abbreviation where it's just a one letter for each amino acid. Glycine would be G, histidine H, and lysine would be K, because L stands for leucine. So a GHK peptide is a peptide of a specific sequence, glycine, histidine, lysine, in that order. It's like words. G-O-D is God, but if you take D-O-G, then it's dog. And God and dog are just not the same thing. So here we have the proof of the difference or the importance of sequences in a peptide and of knowing the sequence, because the peptide glycine, histidine, lysine has been shown to stimulate collagen synthesis in fibroblasts. You put this peptide into a Petri dish with some dermal fibroblasts, and you will see an increase in collagen as a function of quantity. But if you take the peptide and you make glycine, lysine, histidine with a different sequence, just replacing lysine and histidine in their sites, then it's GKH as a peptide, and that does not stimulate collagen. It stimulates lipolysis in adipocytes, in the fat layer. So just a switch of two amino acids in the position has a dramatic effect on the efficacy. Now that, of course, is not something that you can predict, because, well, it's just not possible, not yet. Maybe someday, but that is empirical. If you want to find out what a peptide does, you synthesize it, you synthesize several variants of it, and then you test it in a discriminatory in vitro model or gene model to see, does it do something? Is it safe, or is it active? But remember that the idea of sequence is extremely important to know what a peptide, what you have, and what a peptide will do. The advantage of these oligopeptides, well, of course, we know that there are many active ingredients on the market, have been developed over the last 30 years. And of course, retinol is one, and resveratrol, and plant extracts of all sorts have been developed. Vitamins, of course, are important, fatty acids, ceramides. But what's the big advantage of oligopeptides? Well, contrary to especially the so-called natural products where you have some extracts, but you don't really know what's inside, even if you have some fingerprinting of polyphenols or flavonoids, but it's not the same thing. Here you have a perfectly analytically characterized product, which is, first of all, a safety aspect. You know what you get, and you can reproduce it every time. Reproducible. In a reactor with analytical features behind it, so you know what you get. The peptide is described and identified, and it will have specific biological activity, as we've just seen with the glycine, histidine, lysine, and glycine, lysine, histidine example. A few peptides have sometimes 2 or 3 different activities, but most of them are essentially specific for one chosen activity. And their high potency, they're very, very powerful, which is important. You can use very little of them. That is important from an economic point of view, because peptides are difficult to synthesize, costly. A peptide can cost up to €1 million per kilo. This is rare, but 100,000 is absolutely usual, and no big deal. So peptides are very expensive, but if you can use them at very low level, and we've seen this in a slide before, 3 ppm give you a good result, then they become economically viable because they're so high potent. And using such a low dose means also relative an additional safety aspect, because once the peptide has done its job, it is very quickly hydrolyzed, metabolized by skin-specific enzymes, MMPs and chymotrypsin and whatever, similar proteases, and so it does not get any far into the bloodstream and will have no systemic effects. In addition to that, to the scientist, peptides are fun because you can do quantitative structure-activity relationship studies. You synthesize 50 peptides and try to find out why and how, what they do, and what is their specificity. This allows many more peptides to be found. If you just take a peptide with 5 amino acids, let's say, that's a small, not difficult to synthesize, and you can have 3.2 million different peptides with just 5 amino acids. That is statistical, that is easy to calculate. So 3.2 million peptides to test with just 5. And if you take a peptide with 6, then you have 6 times 3, 18, almost 20 million different peptides. And some of them will do something and others will not. So peptide use in cosmetics has been, over the last 25 years, increasing importance. And now we find in the Inc. dictionary about 1,600 of these synthetic peptides. Only about 50 have found widespread use. The reason, of course, is, as most of you know, the Chinese legislation, the ICIC list, the positive list, which says that only ingredients that are on the list are allowed in a cosmetic product is sold in China, and there are only 66 peptides listed in there. Any new peptide would have to go through this legislation, and that is a very complex and expensive process, so won't go into this. But these about 50 peptides from different suppliers have found use and recognition. Most of them are still used in tissue repair, such as anti-age, the wrinkle reduction, firming, stretch marks, puffy eyes, dark circles under the eyes, etc. But there are also a few peptides that participate in slimming activity, skin whitening. There are not many because it's very difficult to do. What's going to be careful. There are some trends to make peptides with tanning activity. It's a small market, but possible. There are some on the market. There's a decrease in hair loss or even stimulating hair growth peptides. There's recently a few peptides, at least one, that claims and has shown to reduce the loss of color in your hair, so making hair grow again at the right color, not coming out gray or white, but dark, which is very nice. And there are peptides with anti-inflammatory activity, soothing, calming, and some of them are called neuropeptides because they do neuroscience acting on nerve cells. And I'm sure you can imagine further activities with these 3.2 million pentapeptides or more. Now, one thing, of course, one objection to the peptide story at the beginning was, wait a second, only 2 or 3 or 5 ppm? That sounds incredible, that so little can do so much. Well, with the clinical studies, with in vitro studies, ex vivo studies also that you can do on 3-dimensional skin models, it was simply a matter of demonstrating that these peptides work. All right, but how does the peptide get into the skin? We have the stratum corneum, where there's only 13% of water between the layers, and the rest is lipids, ceramides, and free fatty acids, and whatnot. A peptide is, especially these small peptides, is hydrophilic. It has a charged NH3 plus group on one side, and the carboxyl minus CO minus group on the other side of the carbon, charged molecules, and with side chains which contain hydroxyl groups, like in serine, threonine. These peptides are water soluble. So why or how should they go through the skin? I had these objections in the 90s saying peptides cannot work in a cosmetic product. That doesn't make sense. Well, in a way, that's true. And so one has to find tricks to get peptides into the skin through the stratum corneum to the epidermis, and if possible to the dermis and hypodermis if you want to do slimming. So one of the possibilities is to use penetration enhancers, various glycols or surfactants, but some of them have side effects, irritation, destructuring of the lipid bilayers. You can try to use delivery vehicles with liposomes or niosomes or ethosomes or any other somes, or with nanoparticles and nanovesicles. That has to be tested on a case-by-case basis. Not every peptide works with these. Some do, some don't. And of course, there's also possibility, and that has been adopted by most. I must admit, we were the first and patented this some time ago, to add the palmitoyl chain, a C16 chain, to the peptide whenever possible, and have the palmitoyl chain as a locomotive to draw the peptide through the lipid bilayer and all the way to the dermis. By doing studies with peptides, one can show the better penetration with such modified peptides. But again, it's a case by case. Sometimes smaller fatty acid chains, acetyl may be sufficient. Sometimes you need liposomes or other delivery vehicles. Here is just an example, the proof that palmitoylation helps to increase the epidermal penetration. We did a study with the carnosine, which is just 2 amino acids, beta-alanine and histidine, a molecule that we find in our body, in the muscles, and that on the skin just doesn't go anywhere. It is too hydrophilic, too charged, and doesn't go anywhere. We did this on front cells on the skin and looked for half an hour, 3 hours, 6 hours, radio labeled so that we can follow it all the way through the skin. And we compared this to a palmitoyl carnosine, the same peptide, but adding this palmitoyl chain, which is symbolized here, a long C16 fatty acid chain, which is lipophilic enough to draw the peptide into the bilayer. And as an example, the small peptide without the palmitoyl. And here you see the bioavailability. The grafted long chain improves the diffusion up to a factor of 100 and 1,000. It reaches the dermis, but does not go into the bloodstream. It stays in the dermis where it will be active. So these peptides have been used now for all sorts of activities, most of it, as I said, for ECM, anti-age, anti-wrinkle, where you have collagen and GAG synthesis from various suppliers, the Matrixyl I mentioned, but also other companies who have developed their own peptides. As we said, there are so many millions of peptides that can still be made. There is lots of possible research still for elasticity, for stem cell improvement or protection, for melanogenesis, product from Spain, product for increasing lip volume was developed. And more recently, the present trend of neurocosmetics, where you have Botox-like claims of Botox-like peptides, peptides that have soothing, anti-inflammatory activity and anti-irritant activity, usually like the calmosenzyme peptide from Derma, the argireline peptide from Lipotec, the Cinec peptide from Pentapharm. They're all based on peptides that we have in our body, in our systems, but that with our tricks of getting the peptide in and maybe slightly tweaking them, we get them to work to boost the activity of anti-irritation, of muscle relaxation, etc. Getting inspired by what is already in nature has the advantage of reducing the risk of toxicity, because, well, if it's a peptide that you have in your body anyway, there is little chance that will do any harm. And then you can have peptide combinations, synergies between peptides. We look at the pouches, the puffy eyes, dark circles, stretch marks, etc. Even hair growth, hair loss prevention, eyelash growth, etc. are all susceptible to be treated with peptides. You want to know a little bit about the safety and the regulatory aspects of peptides, because I've heard very often, wow, if peptides do so much, and if you talk about the peptide acting on a ligand, this is a pharmacological activity, so these are drugs. This is not you can't use it in peptides, in cosmetics, sorry. Well, no. If that were a problem, all these peptides over the last 25 years would not be in the creams, would not be allowed, would have not gotten there. Peptides, of course, can be used, and peptides are allowed to be active. It is only the way you intend them, and especially you intend your finished product to the consumer. What you tell the consumer, how to present your product and what it does, is important and makes the difference between a drug and a cosmetic. To simplify, if you say this is an anti-wrinkle cream that reduces wrinkles because there is a peptide that stimulates collagen synthesis, then the FDA will pounce on you and say this is a drug claim, and it is true. If you say this is a product, a cream that reduces the visibility of the skin by smoothing the skin, then that is fine. It is a physical effect. The visibility of wrinkles is lowered, is decreased, and you have smoothed the skin, and you don't talk about the biological effect, and that's it. And the FDA and other regulatory bodies will not have a problem with it. The fact that something is active and does something in the skin does not make it a drug by itself. It's only the language of the claim that is important. So we can distinguish cosmetic and medicinal products in this way. FDA, of course, in the US is special. You have to learn how to claim your products. But in Europe, we have this interpretation of the highest jurisprudence, the highest courts of the EU, which came to this conclusion that the capacity to restore, to correct, or modify physiological function should not lead to the classification as a medicinal product by function. That means the fact that the peptide or any other active does something, has some biological activity in the skin, does not make it a drug immediately. Even if it has an effect on the human body, as long as it does not significantly affect the metabolism and the modifiers does not strictly modify the way in which the body functions. So a peptide or resveratrol or retinol or vitamin C, they all have biological activity. They all get into the skin, and this is perfectly legal. Only the way you talk about the product when you make medicinal, therapeutic, preventative claims, then it becomes a drug, not because it contains an active ingredient. That's about regulatory. Safety, very important after 25 years, almost 30 years of using these products. We use very low concentrations. And once the peptide has done its job at the membrane receptor of the cells, it will be rapidly degraded into free amino acids by local enzymes. Most peptides are used based on sequences that we found in the body anyway, and so they possess natural mechanisms of action and therefore pose no threat. There is limited transdermal penetration. Peptides get through the epidermis into the dermis, only a certain amount of what you put on the skin at the top, but sufficient, especially cumulative over time when you use it longer. And nothing gets into the bloodstream. Very, very little, insignificant amount, which don't have systemic effects, don't have effects on other places on the sites that where you have used them. So, of course, if you develop a new peptide and you want to know what it does, first you do both safety and efficacy studies. Classical safety evaluation is, of course, necessary and recommended, especially if you start modifying the peptide or if it becomes a little bit bigger, and then you have to watch out for sensitization. So you cannot immediately say peptides are guaranteed safe. You have to do your studies. But most peptides, those peptides that are on the market, they have this guarantee. So it's practically 30 years of usage. Our first one was developed in 1990. And these peptides of synthetic origin, I'm not talking about the simple hydrolysates of collagen or elastin or milk protein, but these synthetic peptides with a defined amino acid sequence, with a defined and proven documented biological activity. They are safe, they're cosmetically efficacious, not intrinsically incompatible with regulations worldwide, and endowed with a great potential for innovation. You can still make many more peptides and make people excited about them. Of course, if you use them correctly, if you test them appropriately, and if you claim them with moderation. And that is the essential stories of peptides. I don't have, unfortunately, the time to go through many examples because some of them are really fantastic. The pathways of how they work on the DNA, on transcription factors, on various receptors. The peptides have fascinating biology and mechanisms, and I encourage everyone to study this, to test, to run interesting molecular biology, biochemistry studies. You can have real fun and find exciting molecules. And with that, we come to the home messages. We have targeted biological activity, high specificity according to this key lock model I explained, high affinity to the cellular targets. That's why we need so few amounts of the peptides. A hormonal mechanism, even if we don't use the word hormone, because the authorities usually don't like that. They think of estrogen and testosterone, which are indeed not allowed in a cosmetic. So don't use the hormone. Actually, hormone is just a Greek word for messenger. So we can talk about messenger that trigger intercellular cascades. They're stable, they're safe, they're legal, and they have huge potential for further innovation. Thank you for your attention, and I'm ready to take questions.
Well, thank you so much for that enlightening presentation, Karl. That was excellent. We had some great interaction online here, and we have a number of questions. So we're going to just get with those questions. We got about 15 minutes for answering questions. We're going to the top of the hour, but we'll just start out. There were a few formulation questions. So the first one was, when you are formulating with peptides, when should you incorporate them in terms of the temperature, and what phase should you do it, like the oil phase, water phase?
A peptide, when it is synthesized in a reactor, in the end, it comes out as a white powder, very expensive white powder. So using a peptide as a powder at the level of 3 or 5 or even 10 ppm in a cream would be impossible to homogenize. Therefore, all suppliers of peptides, the suppliers of active ingredients, as you all know them, these suppliers, they synthesize themselves or have them synthesized somewhere, and then prepare peptide solutions which are convenient to use. Most of these peptide solutions contain, let's say, 100 to 500 ppm, 0.05 to 0.001 percent of peptide, and the rest is some excipient, water, glycerin, butylene glycol, whatever, or an oily excipient. So you have an easy, convenient solution, which is then to be used in the final formulation at a level of, let's say, 1, 2, 3%. That is easy to stir into an emulsion or a gel that is being formulated. Peptides are stable, as I said, up to a point. It is recommended to incorporate these peptide solutions in the final phases of the emulsification, that's to say when temperatures come down slowly to 40 degrees C approximately, and to integrate them at this time, then there will be no problem. Now, there are exceptions to this. When you make a lipstick, and a few lipsticks have used peptides. I mentioned one peptide as lip volumizing. Lipstick, of course, has higher temperatures, and then the question is, does the peptide survive this temperature? That, in some cases, needs to be verified analytically, but yes, our experience is that even at 80 degrees C, for the short time of making the lipstick, the peptide survives. So if you
I would think it takes a lot of energy to break that peptide bonds.
Peptide bonds are usually broken either by a strong acid, very strong alkaline, or by enzymes. But heat alone, especially in the absence of water in a lipstick, does not do much to the peptide. So, otherwise, if you have a simply hydrophilic gel or so, then the peptide solution is easy to incorporate. And if it's emulsion cream, then one adds it after the emulsion step into the final formulation as the temperature goes down.
Someone asked about when working with peptides, what's the pH that you would want to incorporate them at?
Like most cosmetic products, you want to stay between 5 and 7, but 4 is still acceptable. If you go with very strong acids, then that depends on the peptide, and it depends on the formulation, on all the other ingredients. If there is a risk of slow hydrolysis, I think down to 3.5, peptide will still be reasonably stable. Beyond 8, you would be careful. So best pH is between 5 and 7.
Gotcha. And what is the recommended percentage of peptides in a formulation? I know you mentioned a few times in the presentation they worked at low levels, but what is your recommendation on that?
There cannot be a general recommendation because each peptide has its own activity and affinity. That recommendation is given by the supplier, those who have done their in vitro studies. Usually in vitro studies are done with a little bit of dose dependence. You study with, let's say, 5 ppm, 10 ppm, 20 ppm, or up to 500 ppm if it's necessary. As long as it's economical, that depends on the cost of the peptide, you can do dose response. And then you choose maybe doing an ex vivo study and to see about the penetration, how does it go through, how far to the dermis, to the hypodermis, and that gives you an idea at which concentration level you use the peptide in a clinical test. And again, one has to be precise about talking about peptides, the pure peptides or the peptide solution as offered by the supplier. These are usually 1, 2, 3, 4, 5% of the solution, but one wants to know how much ppm of peptide in the final product, and that would be, let's say from 3 is probably the minimum, all the way to 100 ppm in the final product, rarely more.
Yeah. Here's one interesting one. Would you say that the use of a synthetic peptide is more beneficial than, say, a natural peptide?
The answer is very clear. We have to define what we talk about. A peptide, as I have shown, is a specific structure, an amino acid attached to another amino acid. You can put down the formulation on paper, and every chemist knows what it is. That is a structure, and this structure will have a specific safety profile and a specific activity profile, or not any activity, but if it has an activity profile, it will do something under certain circumstances, this particular structure. And if you are able to isolate an identical peptide from some plant, but really identical, the same structure, the same atoms, carbon, hydrogen, oxygen, the same place in the structure, then that peptide will do exactly the same thing as a synthetic peptide. There is absolutely no difference. Of course, same chemistry, same story. Of course, that is rarely the case, because to isolate the peptide from a plant, even take a plant protein and hydrolyze it into smaller fragments, and then fish out the one peptide sequence that you're interested in. Theoretically, it's possible, but economically, it is just impossible, and nobody does that. You could also use GMO plants to synthesize a peptide and give you the same, or you can do the peptide synthesis rather than in a reactor with specific chemicals and organic solvents. You could use fermentation technology to make a peptide. But again, this is a very complex procedure, and so-called enzymatic synthesis of peptides is a curiosity of the laboratory, but not economically viable. So there is no difference between a natural peptide and a synthetic peptide, as long as they are the same. If the natural peptide has a different sequence, it will be different.
Another question was, is there a peptide that is used for the discoloration of hair? I think
indeed,
that's related to gray herring or something.
I can say the company Lucas Meyer was the first to come up with a peptide that used on the scalp for a certain amount of time. Of course, you use some lotion containing this peptide used on the scalp. The hair that grows out will be less white, less gray, and will retain some of its natural color. That has been shown by one peptide from Lucas Meyer. Other companies, as I've seen, it's become a trend. In the meantime, they have found other ingredients that do that too. Now, which is better, I don't know. I don't compare. But yes, there is a peptide that does that.
Okay, thanks. There were a couple of questions about Matrixyl. Have you seen any major advances in developments since the launch of Matrixyl? And does Matrixyl really work better than retinol?
Over the over the 20 years that Matrixyl exists now, the Sederma company has developed further variants. There is the original Matrixyl, then there is the Matrixyl 3000, which is probably the most used one. This is actually synergistic combination of two peptides. And then there is Matrixyl Synth 6, which was the follow-up Matrixyl that has the capacity to stimulate not only just collagen, but 6 different proteins in the cells which are necessary to do extracellular matrix repair. And then the last one came out a couple of years ago called Matrixyl Morphomix, which is again, let's say, upgrade. Although one shouldn't say upgrade, none of them are better than each other. They're all very good, and the choice of one or the other depends on marketing brief and what kind of specific mechanisms you want. So there is at least 4 or 5 variants of Matrixyl on the market, all from Sederma. What was the second part of the question?
That was in a comparison to retinol.
We did once,
Matrixyl versus retinol.
At Sederma, a clinical study to look Matrixyl 3000, Matrixyl 3000 against retinol, and it turned out that, well, first of all, Matrixyl is 3 ppm, retinol is 700 ppm. And 700 is just the limit before you start getting irritation and consumer complaints. So the amount of retinol or matrixyl that you use is not the same. First difference. Second, in the efficacy in that 2-month study, 4-month study, we saw a quicker effect with the matrixyl compared to the retinol. After the 4 months, the two were essentially the same, but matrixyl worked quicker. And the other difference, of course, is that retinol is not stable, and retinol is at higher concentration irritant, and retinol, after all, is known to be teratogenic. So that is not necessarily the best profile. If retinol had not been called vitamin A, or if you wanted to register today retinol as a cosmetic ingredient, you'd have probably troubles with that toxicology profile. Not stable, irritant, and teratogenic. So there is no no comparison between the two.
Well, it looks like we are at the top of the hour. I will. We've got a bunch more questions, but I'll just give you one more question. And sorry if we didn't get to your questions. I'll compile the questions, and maybe we could do a follow-up then. But here's the final question. You mentioned that there are over 1,600 peptides in the INCI dictionary already, but there is still room for innovation. In what areas do you see the most potential for new and effective peptides?
Well, my glass crystal ball is the same as everybody's. Predictions are difficult, especially about the future. So where can we do something? I have gotten many times the question, well, peptides are very nice, but the matrixyl, of course, was the first peptide and probably the first active ingredient of importance that was studied for 6 months. That was the breakthrough, doing a 2 and 4 and 6 month study and having these great effects. So people said, well, okay, this is nice, but 6 months, God, marketing wants something in 6 hours, in 6 days, and 6 weeks, but not in 6 months. Well, it was still a fantastic breakthrough. But people would like to have an effect quicker or a stronger effect. And my answer to that is actually, well, you can try, but you got to be careful. The skin is resilient. The skin accepts a lot, but there are limits. If you have something that is stronger, let's take retinoic acid, 10 times stronger than activity than retinol. But you know the effects of retinoic acid, and it's not allowed in a cosmetic. So if you had a peptide that was, let's say, 10 times more efficacious than matrixyl or argireline or whatever, you would have side effects. The skin would not allow it, and you would have problems. So better, quicker, faster, I don't believe it. Are there fields where we could do more? Well, stretch marks for the moment has been, a little bit of a success, but that is perhaps a field where you could achieve more important effects. Or tanning is still a field where many patents exist already. Many peptides have been tried, and the effects are not filling the need of what we like. So that is a field where innovation could be of interest. Tanning, and again, this hair loss and hair coloring is also a field where it's a relatively new field, and I'm sure there are other peptides that could do some good job there.
Well, that does bring us to the end of this webinar. Thank you so much. We have recorded this. It'll be up on the replay as soon as we get the video edited. Thank you, everybody who attended. Our next webinar is going to be in 2 weeks with Dr. Nava Diana, and she will be talking about the skin and skin microbiome and issues related to that. So should be a good one. Thank you, Karl, and please stay safe there in France.
Thank you. Sure. Goodbye.
Okay, thanks, everybody. We'll get started in 15 minutes. We're just waiting for our speaker to come on. Hello, I'm Perry Romanowski. I'm the chair of the Education Committee for the IFSCC, and I'm so glad that you're here. We've got a really excellent program today. It's about peptides in cosmetics by one of the industry experts, Dr. Karl Lintner. Just waiting for him to come online. We are not located in the same place as most people are, sort of shut up in their homes at the moment around the world. Karl's actually located in the EU, in France. I'm located here in Chicago. Actually, if you're here, welcome. Please say hi in the chat. We want to get a sense of where people are from. So if you could say hello and put it in the chat there, that would be great. Karl, hello.
Hello, how are you?
I'm good. I'm assuming you can hear me okay.
Yes, I do.
All right. We see you. Looks like everything's working out so far with the technology. We've got about 30 people already online. So
good.
Let's see. I'm looking at my signal. Seems to be doing all right. You never know with video, so sometimes we might cut the video out if the signal's not strong enough. The audio usually will go through perfectly fine. And when we're doing static slides, those will work fine too. Our first person saying hello, Marilo from Spain. Hello, Marilo. Thanks for attending. Just at the moment, I'm going to share a slide. Where is that slide? There we go. Okay, hopefully you can see that slide and still see us. So if you're there, we've got Arwa. Hello, Arwa. We're going to get started. Rosa from Spain. Hello. A couple of people from Spain. That's great. Shivati from India. Hello. Hello. Welcome. Well, Karl, this is an exciting topic we got here, but since we have a few minutes, I just had a couple of questions we might just ask. How did you end up getting into the cosmetic industry?
That is a good question. On a very roundabout way, I did my PhD in France in a nuclear research facility, huge structure with 10,000 people and so on, in a laboratory where there was a group, an international group, and among them was also one French student who had done his PhD on peptides, and I followed in his tracks and did my PhD also in peptides. And then he left soon thereafter because academic research was not his kind of, well, not his beer, right. Cup of tea, as we say. And he joined a very small company at the time that was a contract manufacturer for cosmetic, small-scale third-party manufacturer. And I stayed on for 10 years in the research facility doing fundamental research on biochemistry, biophysics, biology. And then I got bored and left the academic situation and went into big industry in Germany, where for 6 years I worked in the, well, for the food industry, cleaning and sanitizing, doing everything to make food safe. That was very interesting, of course.
Yeah.
And for various reasons, I came then back to France and a new job, and that didn't work out. And my friend, who had been in the same laboratory in the research facility, he said, "Oh, you're back in France. My technical director just quit. Would you be interested?" And I was, because the previous job hadn't worked out. So I said, "Well, let's have a look." And I was a little bit skeptical. Cosmetics, I was a scientist, right?
Yeah, I had that same feeling when I started in cosmetics. Yeah.
Right, especially as in the 80s and 90s, most of the ingredients were animal-derived, cow brain extract and cowhide and snail mucus and whatnot. I didn't think that very serious. But actually, there was a crucial moment because in 1990, this BSE, the mad cow disease, cropped up, and the industry that reacted most quickly, scared.
That's right.
The industry, they didn't want to have anything to do with animal extracts anymore. At Destillato, there was this paper on the door for suppliers saying, no fish, no milk, no animal. If you don't have anything else, go away. So our company, and my friend asked me to join, and I said, all right. I saw the company that he had created in the meantime, very nice facilities. He had the first intra-company network and computers all over. So I became a technical director of this company. It was an interesting change because the big food sanitation company that was Henkel in Germany with 33,000 people, and now I joined the company with 33 people.
That's a big change, yeah.
I was the technical director and scientist, and the boss was the owner, my friend. So we got along very well. He was the sales and marketing specialist, and I was Karl, the science. And so my job was, of course, very quickly to develop a whole new product line to replace the animal extracts by something different. Plants and biotech, and well, that's where the peptides came in, because his and my PhD had been on peptides, and we thought, well, this is an idea that has not been tried yet in the cosmetic industry, other than just hydrolyzed proteins. But synthetic peptides with specific activities, that was new. And so we tried that, and that became a very good success for the company, for me, for everyone.
It's interesting that there's still this consumer perception that animal ingredients are still used a lot in the cosmetic industry. And you're just evidence that's really just a misperception by consumers. I think most animal ingredients have been taken away, except maybe lanolin or something like that.
Right. Even lanolin had big problems for a while, not only because of animal, but also because of this wrongly accused irritation and allergy story. Mm-hmm. But yeah, even today, there is, let's say, reticence to use animals. Yesterday, I talked to somebody about silk proteins, and they said, we can't sell it. People consider this as animal, and we don't want it.
And beeswax even is a problem too, yeah.
Which one?
Beeswax.
Beeswax, yes. Well, that is really, well, if you talk about vegan, and vegan is...
vegan is... That's sort of a new trend these days, that vegan is like, wow. Get rid of a lot of good ingredients.
Exactly, which is a shame. But at least they are not against synthetic ingredients, which is an advantage. Because other people say, oh, if it's chemistry, I don't want it either. So, but yeah, some animal products are making a comeback, like the snail mucus. Now they call it, I don't know what, but some people have shown some benefits, additional benefits to what we have shown. And snail mucus is back. You see a little bit of keratin, and hydrolyzed fish collagen is still being used, but otherwise very little. No, there's not many animal anymore.
All right, looks like we've got a lot of people signing in. That's great. We've got over 75 people already. Welcome, everyone. A lot of you have found the chat. If you haven't found the chat, there's a little chat button there. Feel free to post where you're from, who you are. And during the presentation, what I want you to do is, if you think of questions, feel free to post your questions there. We're going to collect all the questions, and at the end of the talk, Karl will be around to answer some of those questions. So it'll be interactive in that way. Boy, we've got people from London, we've got India, lots of people from Spain, New York City, hello New York, Mumbai, India. Wow, Karl, you are internationally famous here. It's going to be an excellent talk. Now we got just 5 minutes. And Karl, you've been in the industry a pretty long time before we start here. Do you have any advice for somebody new starting out in the industry? Do you have any advice for them as they get started on their career?
Advice? Oh, I don't want to be pretentious. I've indeed 30 years in this industry, and I've enjoyed it very much. And that is important. You're going to have fun, and fun comes from creativity. Well, the important in this particular business is to be flexible, to have to look for ideas, sometimes even crazy ideas. Never hesitate, but still keep your feet on the ground.
Yeah.
Be polyvalent, not a specialist in one tiny super specialty and to be the super expert. No,
Yeah.
in this business you have to look at everything: production and marketing and sales and of course the research and the science. It all goes together, and it all is important. So polyvalence is something I try to teach my students also, and when they have understood it, they made good careers. So I think that is important. Of course, science, if you are science-oriented, the knowledge is important, but not just that.
There's some fabulous science in the cosmetic industry, but I know when I started working for a company as a formulator, they really try to pigeonhole you. The company wants to pigeonhole you to learn, like, you're the hair care person or you're the shampoo person, and then there are chemists over here. These are the skincare chemists, and you work on specific brands. And from a company standpoint, they like to get you to specialize in a thing. But I think from a career standpoint, as a formulator, you should work on hair care, skin care, color cosmetics, whatever you can. You really want to expand your formulating skills and also your knowledge about raw materials. I'm sure you've had to learn about surfactants, preservatives, peptides, active ingredients, and all these things. There's a lot to learn.
Absolutely. The classes I taught at this special school in France, that is a school for perfumery, cosmetics, and aroma, what I taught was ingredients. And I told them a cosmetic product is only as good as the ingredients that you use. So if you don't know anything about the ingredients, you can do all the science, molecular biology you want. Your product will not be of interest, will not be good. So somebody who knows the ingredients has already, and everything that goes with it, the sourcing and the toxicology or the safety of the ingredients and the compatibility and whatnot. The more you know about the ingredients, the better you can understand and work in this industry.
Yeah, excellent. All right, we've got just under 2 minutes. We've got people from France, from Finland, from Iran. Hello, Belgium. Wow. Lots of people from Spain and Barcelona. I do hope to get to Barcelona someday. I've never been there. From the Netherlands, we've got Maria from Mexico, Italy, Sylvia from Italy. Wow, we've got people all over the world, Karl. We've got almost 130 people already online. I just want a little housekeeping before we get started. I am Perry Romanowski. I'm the educational head of the educational committee for the IFSCC. This is our second IFSCC webinar, and it's a new thing we're trying out here. So we're getting experts on topics of interest to cosmetic chemists around the world and sharing with you, our members. Today's expert is Dr. Karl Lintner. If you have questions while he is presenting, feel free to post them in the chat. If you have any technical issues, one of the things that can sometimes happen is that the signal will start to get choppy and come out. You're going to just hit the refresh button on your browser. That usually will fix any problems. Karl, if there's a problem with the sound and I break in on the video, you'll know, and we can work that out there.